Okay, student T test, square test. Okay, this is just different. Right. So just I have to brief about these two tests. We have to just try to understand these two tests. And okay.
Sir, I read the worked examples and I found it hard to understand them.
Which one? The above one? This one?
Yeah, yeah, this one.
Fine. So this is just table, right? They have collected the data. What is the data? What is the data for? So fine. So there is a like hypothesis. An example number one. Okay. So after the new volcanic island of Surtsey was formed in 1960, scientists observed that colonization of the bare lava. Now you will analyze some data of the scientists collected about the abundance of the species there over time, right? So due to the volcanic eruption, formation of land taken place, and after that the formation of the land, the plant grew up, and then after thousands of years, plant grew up, and then species, different species, animal species came over there. They colonized. Now the scientist is studying about some data. What kind of data? They are studying the data for the this one species diversity. So look, year year after formation, right? Now this is the year 5, 10 year, 15, 20, 25, 30, 35, 40 year. Species total 4 species, 10 species, 13 species, 11 species. So these are the number of the species. This is the number of the year. The total number of the species in search over the time, right? So this is the number of the species which came over there uh, uh, after number of the species, right? Now the null hypothesis is that there is no correlation between the time of year and Surtsey was the first formed, and then the number of the species present. Rank variable, calculate the d after d square. Okay. So then what they make a null hypothesis that there is a no correlation, right? That is a negative null hypothesis. Okay. Come to the next. Now there is a variable x variable, the number of the species, ranking of value x from the lowest to highest. So that is the ranking, right? Lowest one, number 1, 2. This is nothing, just ranking, right? 1 to 9. N, so total number of the value 1, 2, 3, 4, 5. So n is the total number of the values, right? Now y, what is the y? Y value year after year formation in, right? So how many year taken place formation? So 5, 10, 15, 20, 25, 30, 30, 30, right? Now the ranking of y values from lowest to highest, right? 1, 2, 3, 4, 5, 6, 9. So that is the total value n, right? Now variable. Now what they did, this is the sum of d square. Now what is the d? What is d? That is the, can you tell me what is the d? Yes.
Isn't it the difference?
Exactly. That is the difference, right? That is the difference. So they got the d, and this is the sum of the d, right? This is the symbol is sigma. That is the sum of the d square, right? So d, that is 2, right? So the sum of the d square is 2. So that's how they got. Now the same formula which we studied over here. This formula, right? Rn is equal to 1 minus 6 d square n, that is a number, n minus 1. They have taken this same formula. Now this taken the same formula, they put the value of the formula, right? They put the value the same.
Sir, how did they get 2 1s for difference?
Sorry,
D squared. There
d
are
square.
2 1s. Yeah, in the last. Yeah.
Okay, in the last.
There are 2
Okay.
1s.
Fine. Look, 1. Just. Oh. uh, there is a difference of the difference in the rank between the pairs. This is the fourth. Look, this is the fourth rank, right? And this is the
Yes.
fifth rank, right? So there is a
Yeah.
difference between the rank, right? So
Yes.
here the 4 minus 5 minus 1. Got it?
Uh-huh.
Now here the 5 minus 1, 1, right? And whenever you use this symbol of this sigma symbol, then
It means total.
total, right? And what is the square? D square. So 1 square is equal to, when you make the square, minus 1 is become plus. Clear?
Yes.
Minus 1 square is equal to 1. 1 square is equal to 1. So 1 is equal to 2. So sum of the d square will be 2. Is it clear?
Yes.
And they put the same thing in this formula, right? And now the value is 9.
Sir, why is like 9 multiplied 80? Where did the 80 come from?
Okay, 9 multiplied by 80, just a minute. So the n minus n square. So what is the value of n? N is 9, right? N square value 18,
81.
right? 81. 81. So 81 minus 1 is equal to 80,
80.
and n is 9.
Okay.
So 9 into 80, right?
Uh-huh.
Same thing is above. D square is equal to 2. So 6 into d square, right?
Mm-hmm.
Same. Minus 1 is here, minus 1 is here. The same value have been put over here, right? 6 2 is 12, and here 9 8 is 720, right? They just simple. It's a simple calculation, nothing else. Got
Okay,
it?
what about the last table? That's completely confusing.
Okay, okay. Now you need to use a critical value table. See the table C. So you need to accept your null hypothesis. So our value is 0.983, right? And if extract from the table critical value Superman's correlation coefficient P is equal to 0.05. Okay, where is our look? Now what we studied, we have to go back. So this is d square. Look, in last class we studied, find the probability of value related with your observation value in the pairs. Ignore where R is positive or negative. If the value is less than 0.05, the correlation is considered to be statistically significant. So that so you can reject your null hypothesis that there is a no correlation.
So if it's like less than 0.05, if the values are less
Right.
than 0.05,
Right.
they're
If-
rejected.
Exactly. And if it is more than this one, then the value will be, then your null hypothesis will be accepted. So your value
That
is
means
0.
there is a correlation.
Exactly. You
But
got
what
my
if all of the values are above 0.05, but one of them is less than 0.05?
point. This total value, RS value. So what is the
Okay,
RS value?
RS value.
Exactly. So RS value
Okay,
is
so
it's
RS
more than
value has to be
0.05.
above 0.05.
Exactly. So there is a correlation between the two. That's it.
So what's the point of the last table, table C?
This table you will get this table have been given. This is the standard table actually. This is the standard table. You need not to make this table. You will get this table in question.
Okay. What are critical values?
Critical value is you can say that is called the standard value. Means what should be the standard, like if the number of the means at particular rank, what should be the critical value, what should be the passing marks. Got it?
Okay.
Now come to the another student, another boring concept. Student t test, right? The student t test is used to determine whether the mean of the variable in one group differs significantly from the mean of the same variable in different group. right? Got it or not got? No.
No.
There are two teams. One of the
Okay.
team, in one of the team, there are, irrespective of, leave the game because I'm not a sport lover. So leave any game. So there are two teams. Each team have the 10 members, right? And suppose that they all are batsmen. So like in A team, the first man have made the 2 run, then the second one made the 20 run, third one 40, 50, and like all they made the runs, right? They all 10 member made some runs. The same way in the team B, all 10 members made some run, right? Now we have to calculate the total runs of both of the team. Then we will get the, and we will divide by 2. So we will get the mean value, like what is the average mean run they made, right? Now that is the when then what we are taking, we have to compare the average run of both of the teams. Got it?
Okay, so you take out the mean for each team.
Exactly. Right.
So you
Okay.
add the number of runs made by one team and then you divide it by 2.
So that's we are taking the mean value of both of the team. Then we are comparing the mean value. We are not comparing individual. We are comparing only the mean value. Got it?
Yes.
In this test, the same kind of situation is over here. The student t test is used to determine whether the mean variable in one group differs significantly from the mean of the same variable in different group, right? For example, comparing the blood pressure of male and female is stated simply. It tests whether the two tests of that data are significantly differ from each other, right? So average mean blood pressure of male and female, does they differ or does they are similar, right? The variable being considered must follow the normal distribution. The student's t-test considered the degree of overlap between two sets of the data. Allow you just whether they are difference between the mean value or two groups statistically significant just due to chance, right? So t-test will tell you that either they are like matching with each other or they are not matching with each other. Got it?
Yes.
So if what is the difference between, either there is a similarity between the mean value of two or there is a difference. So how you will just, if you have to just the similarity and difference of the mean value, then you have to go for the t-test, right? Okay.
Okay.
Fine. State the null hypothesis. The null hypothesis that there is no significant difference between the mean variable for the two categories, right? Even you can make the null hypothesis, yes, there is a difference between the, there is significant difference between the mean value of two, right? So you can take positive or negative null hypothesis. Calculate your observed value t, right? Now what is the t? You will need to calculate the t. First, you need to find the mean variable s square of both of the group or the sample size. For example, the number of the people in the group and x variable uh, variable in the measured variable. For example, one person's blood pressure. Then what is the x bar sigma x upon n, right? So x bar means x average. That is sigma x means the total number of the values, and n the number of the values means how many digits are, means how many ranks are over here. The same thing is over here. Nothing different. Now you you can, this is very complex formula. X1 average, x1 bar, x2 bar. Now s1 now x1 is the mean first set of the data, x2 bar is the mean second set of the data, s1 is the variance of the first set of the data, s2 is the variance of second set of the data, n1 is the number of the data value first set, same thing, right? Now you have to consider only, you have to remember only variable, right? You will get the variable in the question. Definitely when you will get the data, when you will get the question, if they want you to calculate the same, then you will get the value of variable, that what is the variable over there. Decide whether acceptance or whether to accept the null hypothesis. To decide whether the value t indicates significant difference in the variable difference between the two group, you need to compare the observed value t a critical value table. So you will get the table like the same way you got in the above example. You will need to know the degree of the freedom, right? Degree of correlation means degree of the freedom. Degree of freedom is n1 plus n2 minus 2, the total number. So what is the n1 and n2? We have got already the number of the data value of first data and n2 number of the second value, right?
Okay,
Find the,
so that's the first thing you have to do.
that is the first
Need
thing
to
which
know the degree of freedom, and then you can use the above formula.
no, first you have to use the above formula, then you will find the degree of freedom.
Okay.
The sequence will be the same. Find the probability value related to the observed value. Now we same value and the degree of freedom. If p, right, if p is 0.05, the difference between the means of the two group considered to statistically significant, and if not, due to the chance, right? Now the value is given over here. Look, this is the x1, this is x1 square, this is x2, this is x2 square. Now what is the x1 bar? That is the average x1 bar. So total is 749 and total the values are 10, n is 10. The same here, right? They got the x2, right? 768, then it is divided by 10. They got the x1 and x2. Now come to the s1 square. So s1 square, they have the square of the both one, like x1 square. So the sum of the x1 square is s1 square, right? 56837, right? Now what is the sum of the this one?
So x1 square is square of each x1 value.
Exactly. X1 square is x square of each x value. And what is the s1 square? S1 square is the sum of this value, these values.
Okay, sum of the value.
Right. And s2 square is the sum of these value, right? Okay. Now what they did? This value minus, they put the same thing in this formula, this one, this formula. So the square of this one upon 10, right? The square of this one upon 10. N1 and N2, same formula.
Okay, so that s squared formula, that's the one they use.
Right. Now the same thing they have put, they have solved, but you won't get the question like this way, like to get the value. Till I haven't seen that, you will get this kind of the question. Then again, they got the value is 0.57, right? And 0.57 is it minus 0.057. So sign does not matter, right? And this value is not less than 0.05, right? So what is the correlation? Look, the number will calculate 0.1 is the absolute, 0.0 is for less critical. This is much greater than 0.05. It means that there is no significant difference between the means shell. Therefore,
If it's too big, if it's too
big.
It's a lot bigger than 0.05, then it's
Right.
irrelevant.
Then therefore we accept the null hypothesis.
So what is levels of significance?
What is the level of
Significance, table E.
If this value, what the outcome is less than 0.05, then that will be insignificant. And if this is greater than 0. Means if it is 0.01, 0.02, 0.03, then this is irrelevant.
What values are written of on levels of significance?
Sorry,
like where did they get these values?
From where did you get this? Well, this value will be given to you on the table. You will get this table. You will get this table in question along with your question.
Like why do we get this? How does it help us?
Okay. You have to compare. Where does you lie? Level of significance, right? If it is 0.02, then it is significant. 0.02. Look, all the values are less than 0.041. 0.05. All the values are less than that, right? Then this is the level of insignificance. These values, these are the level of insignificance. But as we do not need it because the value is more than 0.05.
Okay, the value is 18, so it's much bigger.
You will require it if the value is less than 0.05.
Okay, the level of significance values will be given by the examiner, and degrees of freedom, that's something that we will find out.
Exactly. Now you got it.
Okay.
And you will compare your degree of freedom from this table.
Okay.
By the way, what I suggest you, what you will get the question, you will get a question, and there you have to tell that which test you have to apply over there. That's it. And you have to write it down that test. You will get that kind of question. You will get a hypothetical situation. Now you will get the question which formula or which test must be applied on there. Then you have to just catch the word. If it is talking about the average, the difference between the mean, then the test will be t square.
Okay.
Chi-square test. What is the chi-square test? The chi-square, that is x-square test, is the relatively simple statistical method used to estimate the probability that any difference between the observed and expected result due to chance. Means that what is the what is the difference between the observed and expected value, right? Means what is standard value, what the standard value should be. For example, the pH value of any water which we drink should be 6.5. Now you have the 10 sample of drinking water and you are checking so what is the standard value should be 6.5, and you are checking all the waters, and now the pH value of all the water. So what is the difference between this standard value and the actual value? So when you have to observe between the standard value and actual value, like for sake of example, the blood pressure of a normal human being should be 80/120. Now you have collected the sample of 10 students and you are checking the reality. Does it really work or not? Then you have to use the x-square test. This kind of these kind of the test. Now come to over here. The distribution of the blue and yellow flower, or distribution of the insect and different flower. The chi-square test will tell you whether or not your result differ significantly from expected outcome. So chi-square will tell you that does your result differ from the standard value or not. The test involves some formula of chi-square, that is x square. X square is equal to sigma 0 e to the whole square upon e. In this equation, O is your observed value, means whatever the data you have observed. E is expected result, means the result which is expected, means the standard value. And the symbol C means sum of the. State the null hypothesis. Now there is another example. The basic null hypothesis for the chi-square test is that there is no significant difference between the observed value and expected frequency. This is modified to be specific to the data you collected. Calculate the chi-squared using the formula given above, right? Now plant with a round piece, right? The hypothesis round to wrinkled, the plant with this is the phenotypic characteristic, plant with round piece. The hypothesis, that is the ratio of round to wrinkle ratio 3. Observed value 640, expected result 60. Now O minus E, 640 minus 660, right? There the value is minus 20. O minus E square, 20 square, that is a 400. OE minus, so this value we got it. The plant with wrinkled pea seed. the same, we got the value. Now x square is equal to sum of the both, right? Now the actual value 2.43. To decide to whether accept the null hypothesis or not, means this is the final result. First, calculate the degree of the freedom in the system. This means the spread data in this chi-square test. So what is the degree of freedom? N minus 1, right? Now what is the value of n? What is the value of n? N is give me one second. Okay. Now what is the n? Just minute. The first calculate the degree of the freedom of the system and the measure the rate of the degree of freedom. Once you have observed x square, the value of degree of freedom, you can use them x square probability table. Okay, probability table to find whether there is any significant difference between your predicted observed result. This will tell you whether your null hypothesis is correct or not. Whether if same thing, if this is if less than 0.05, the difference between the two groups is considered to be significant, statistically significant. They are not due to the chance. Now what is the what is the total value x square adjustment? Okay, so x square is less than 0.04 or more than 0.05. Tell me, Affaf. X square.
It's more than.
It's more than. So if it is more than, then what is the result? The difference between the two groups is considered to be statistically significant.
Okay.
When it is less than 0.05, but this is more than.
Yeah.
So this is more than 0. Then the difference between the two groups is statistically insignificant, right?
Insignificant or significant?
Insignificant, because it should be the value of x square, that should be the value of p, right? Probability that should be
But
less than
sir,
0.05.
the reader says it's statistically significant.
Where? No.
Here, look at the
Which line?
page 46.
46. Okay. Once you observed the x square value, degree of freedom, you can use the x square probability table to find whether there is any significant difference between the predicted observed result. Okay, so where is the p?
If p is less than 0.05.
Right. If p is less than 0.05, then the difference between the two groups is considered to be statistically significant. But what is the value of r value of p? What is R value of P? Table is
Isn't it 2.4 something?
P is given over here. Look. P value for x square distribution. Then degree of freedom. So what is R value? R value is 2.43, right?
Yeah.
So for 2.43, just for 2.43. So where is 2.43? Then the value will be for us for 2.7, right? 2.43 will lie in this bracket, right?
Yeah.
What is R value? 0.01.
Okay, then 0.10.
Right. And
Okay.
0.01
So it's
is less than
significant.
Right. Statistically significant. This is statistically.
Yeah, this one is given by the examiner, right?
Right. Definitely, you will get this value because you cannot calculate this value. It's like whenever you get the question related with the log table, you get the log table, right? So these are that kind of the tables. You will get the table because without the help of this table, we cannot infer this table. We cannot get this table. The same kind of the question are given here. Example.
Sir, did you? I sent you the exam paper, right, with the mark scheme. Did you find any question that requires us to do
I'll open.
such calculations?
I don't. I didn't find. Just a minute. Let me open it. That's why I'm saying. Question paper. Let's open this question. Look, let me open your question paper in front of you. This is the question paper which you sent me.
Yeah.
Simple reaction based question, straight questions. Diagram based question. Simple straight question. Explain the role of stroma. Stroma.
So I guess this one's easier
Very
because
easy.
they have versions of papers.
Right,
I
very
guess this
easy.
one is easy.
Right. Very easy.
Easy for you.
No, even easy for now. It must be easy for you as well because we have, whatever we have studied, you'll be able to write it clearly. What is the
Yeah,
difficult for both of us? Whatever we have not neither seen and else what we did not understood. That is the only difficult thing. The day you understand, it becomes easier for you. It applies both of you, on and as well as on me. When I am a science postgraduate, so when somebody used to talk about the commerce, share market, and all that, I was really a bit very disturbed, and I hardly get any point what they are talking about. So over the years, I came to know that how does it work, how does it work, and what is all this. Now it's easy for me, right?
okay.
Yeah. The day you get the thing, then it's become easy for you. When a dead animal decomposing area called Cavender Decomposition Island can form. Photograph below the Cavender Decomposition Island.
By the way, sir, the questions that are marked with a star, you have to be very careful when you answer them because they have to be perfect grammatically and spellings have to be perfect. You have to use very scientific terms in those questions.
The question we have, which have been star mark.
Yeah,
This
this
one.
one.
Okay, like this one. Below
Yeah.
the two statement, but Cavender Decomposition Island. Till now we haven't studied it. The formation of Cavender Decomposition Island is an example of succession. Okay, Cavender Decomposition Island increases the biodiversity in area. Using the information in above photograph, the knowledge explain these, but nothing else. It's very easy. These are very easy questions. They have just given the outlines. They have given the diagram, and on the basis of diagram, you have to write out the concept. So very simple. If you know the concept, then you will reproduce it. Entomology, they have not, means the study of the insect. The graphs on result they study. Describe the conclusion that you can make. Chalo. Look, let's let's do this question. A study of predator in marine environment and island environment was carried out. The trophic level of each predator and the ratio of number of each predator and prey predator ratio were determined. The graph is shown below. The broken line, predator in marine environment, and this straight line in predator environment. Like this is the prey ratio and this is the trophic level, right? Prey predator ratio. Trophic level increasing, prey predator ratio is decreasing. In marine, trophic level very steep, it's very steeply, it's decreasing very steeply in marine environment, and trophic level is increasing, the prey predator ratio is decreasing slowly. That is the only outcome, and that's what we have to describe over here.
So we have to write 3 points, 3 differences.
3
for 3 marks.
Sorry,
3 differences in 3 for 3 marks.
3. Okay, we have to write it down, 3 differences for 3 marks, right? So
Yeah.
first, graph inclination, first thing, right? Second thing, that is the prey predator ratio is already greater in where? Marine environment, right? Already greater in marine environment, but in land environment it's really very less, right? It's lesser, the ratio is already lesser. Second, and we can third difference, that as the biodiversity is much more in marine environment, so that we have to conclude by our own logic. The biodiversity is very complex in this marine environment, so what will happen, that the food chain will be complex one, right? And the pre-predator ratio will be very stable. But in the this one terrestrial environment, land environment, the food chain is very not that much complex, and hence the this pre-predator ratio will be less stable. Concluded, that's it. These are the things which you put from by your own logic, because you won't get everything from your graph, everything from your diagram, right? That's how you have to write the question. This is simple question. Not biotech question.
These are yet to be done, right?
Yeah, those are yet to be done. Biotech we will do it, and genetics we do it. This is the PCR technique. Here, look, explain the multiple copies of the mammoth DNA could be produced. And now we know that the multiple copies can be produced of the DNA, can be any DNA can be produced by the PCR technique. You have to write it on the PCR technique. That's it, nothing else. Very simple, straight question. But it's the indirect question. Very simple question. As such, there is nothing complex. Nothing complex. You can attempt the complete question if you know the concept. If you get the complete concept, it seems to be complex, but it's very easy. You have to just study the data, the chart, and you have to explain it. That's it. Nothing. So there is nothing complex of. Just you have to get the concept, and you have to keep this concept in your mind, right?
Yeah.
So this was your book, right? And
okay, so we finished 5B.
Exactly, we finished 5B. Now you have to do what you have to do. You have to like search some questions. You have to get some questions from different the sources you have, right? And then you have to solve that. And in case if you get fail, in case if you are not getting the concept, then you have to send that question to me. I'll tell you that how we need to solve that question. Got it?
Okay.
third book, third PP, third, which you sent to me.
That's 5C. I
5C.
sent it via WhatsApp.
Yeah, I have that in my laptop. This one, right?
Yes.
Climate change. So far, how what else language you use other than English?
I use Urdu or Hindi, as you can say.
Oh,
And then there's Punjabi, which I speak a little bit.
okay, so you can understand Hindi and Punjabi as
Yeah.
well.
Yeah.
Oh, that's wonderful. Okay, producer, primary consumer, secondary consumer, tertiary consumer, decomposer. Right, it's easy. Mm, ecosystem, I think. Okay. So, do you have idea like what are the consumers? What are the like primary consumers? What are the secondary consumers? I'll tell you one by one.
Okay.
Any idea?
Consumers. Yeah, of course, primary consumers are like, for example, deer eating a
Okay.
plant, and then the secondary is the predator,
Okay.
lion eating the deer.
Right. And tertiary, top consumers. Look. And then decomposer. Fine. Look, Affaf. What are the first producers? Plant are always producers, right? So in the producers, will always consider green plant because they are the only energy producer in this earth, which produces
Okay.
food. Rest of the component of the food chain, they just take directly indirect nutrition from the plant, right? Either directly, like deer take directly and lion take indirectly. But end of the nutshell, these are the like primary producer or producer which produces the food and is in this level. So producers are always kept in the first trophic level. You have to understand that producers
Food
are
level.
right. Food level is food chain, right?
Yeah.
Trophic level is food chain, right? So plants are always at the first trophic level, right? Then those animals which eat plant and they are herbivore basically, right? So plant eating animals are herbivores. They comes in the second trophic level, right? So primary consumer always comes in the second trophic level. Getting my point?
Yes.
And who are the primary consumer? Herbivores. So herbivores will always come in second trophic level. Clear?
Yes.
Then I have written our example as well, like zooplankton and grasshopper. Then who are the secondary consumer? Secondary consumer are those, they will always come in third trophic level, and they are basically carnivores, right? Then tertiary consumer will be always considered top carnivores, like eagle, like lion, right? They can eat herbivores, they can eat carnivores as well, another carnivore, right? So they will come in the fourth trophic level. That is the top. So after the fourth, there cannot be fifth trophic level, right? Please note it down, like man and lion.
You want me to note down this
Sorry.
whole diagram? You want me to note down everything?
Yeah, everything,
Okay.
everything. No, no, just the same pattern, the same in the same.
Okay.
It's written in very vague manner. If in case if you are not getting, you can ask me.
Okay. Done.
Done? Okay, fine. So you got all the trophic level and all that, right? One more important thing, additionally, which I want to share with you. Uh, what is the 10% law? 10% law is, that is, it work only in grassland. 10% law says that when one animal or one organism eat another one, only 10% energy is transferred from one trophic level to another trophic level. For example, if there is 100 calorie energy in this producer, when primary consumer will eat, it will get only 10%. Rest 90% will go in waste. Got it?
Okay, so 90% of the energy
Right, will go
is
in waste.
lost, and
Lost.
10% is received by the herbivore.
Right. And from primary
Okay.
consumer, when it will go away, it will become 1 calorie. Again, 10%
Oh,
will loss.
okay. 10% from the 10% will be lost.
Exactly. Again, from 1 calorie, it will
Okay.
become 0.1. Right. So whenever the energy transfers from one trophic level to another trophic level, then 90% energy get lost. Only 10% is carry forward.
Okay. 10% is carried forward, 90% is lost
Right, right.
in each level.
Exactly. That is called
Okay.
in each trophic level. That is called 10%. Now, let me take what is the next topic.
What is PAR? 2%, 2 to 10%.
Photosynthetically active radiation.
Yeah,
PAR.
but like, why did you write it down here?
We have already studied it in photosynthesis. The light, the spectrum, the total light fall on the earth. Out of this, only 2 to 10% spectrum of the light is usable by the photosynthetic plant. Got it?
Okay.
The total light fall on the earth surface. Plant uses only 2 to 10%. Clear?
Okay. Yeah.
So, what is the next one? Okay. So, food web. Okay, okay. What is a decomposer? Decomposers are never considered in the food chain. Why? Because after every food chain, the decomposer will come. When the animal died, either producer died or primary or secondary or tertiary consumer died, the bacteria breaks down their complex molecules of body and convert into simple molecule. This process called decomposition, and the organism which are involved like bacteria, they are called decomposer. So after every food chain, decomposer comes. So we never consider decomposer in food chain. Got it?
Yeah.
Affaf, audible? Hello? Hello? You are not audible to me. Am I? Hello?
Hello.
Yes,
Sorry,
Affaf.
sir. My internet stopped working.
Okay, okay. Not sure. So fine. Now, what is a food web? When multiple food chain overlap each other, right? It become a food web. When food chain become complex, it becomes food web, right? So the complexity of the food web will decide the stability of any ecosystem. If you want, you can note it down. Please note down this statement.
Okay.
When please note it down. When the different food chain overlap each other, when the number of food chain, when the number of food chain overlap each other, they makes a food web. They makes a food web. Let's stop. The complexity of food web, the complexity of food web will decide the stability of ecosystem. Full stop. As much
Stability of what?
ecosystem.
Okay.
Full stop. As much as the food chain will be complex, that much ecosystem will be stable. Now, what are the ecological pyramids? Now, let me tell you what is the ecological pyramids.
So did you say as much as the food chain will be complex or food web?
Food web, you can say food web, because
Okay.
the food web will be complex. Let me tell you the ecological pyramids. So, what are the ecological pyramids? Ecological pyramids are nothing but they are the just representation of trophic level, right? Whenever we
Sir, are you screen sharing because I cannot see anything?
you are not able to see the screen with the screen I shared. No,
No.
it's due to the poor network. Let me switch off the camera. Is it visible to you now?
No.
So I'll share you again. Now tell me,
Okay.
is it visible to you? Just give me 1 second. Is it visible to you now?
No.
No. Then exit and log in again.
Oh, yeah, I'll do that.
Yeah, actually.
Yes, sir. I can see it now.
Visible?
Yeah.
Okay. So look, this is the pyramid of number. In pyramid of number, we always take the number of the organism at each trophic level, right? So look,
Yeah.
the first example is you have to draw this pyramid, right? Whenever when I teach, after teaching, you have to draw it, right? The first is
Okay.
pyramid of grassland ecosystem, right? Here we are talking about the grassland ecosystem. So in grassland ecosystem, you can see here uh, the first in grassland there are producer. So producer are grasses. So number of the small grasses plant are millions over there in grassland, right? So their number will be larger. So let's say there may be 200 million grass plant, right? 200 million grass plant. Then out of these, in grass plant, the insect which are staying in grassland, their number will be less. Let's say they are only 10 million, right? Insect. Then the insect will eat the grass. Then snake or lizard, which are dependent on the insect or which are eating the insect, their number will be even less. It may be 1 million. Finally, there is a eagle which is the top consumer or top carnivore, which can eat the snake as well as the lizard. Their number may be 0.1 million, right? So ecological pyramid, the pyramid is getting narrow because the number of the number of the number of the organism at every trophic layer is reducing. So this is upright ecological pyramid. Is it correct?
Yes.
Okay. Now the B is another one. This is the B is the pond ecosystem. In the segment of producer, so when we talk about the producer, this one. When we talk about the producer, so at the level of producer, there will be algae or phytoplankton, which are green plant and they make their food. Producer, these algae on phytoplankton will be eaten by the small fishes. So small fishes are primary consumer. So their number will be less than the number of these algae and all that. Then these small fishes will be eaten by larger fishes. Their number will be even less. They will be secondary consumer. Right now, the top consumer will be tertiary consumer. Right, tertiary consumer. They will be, so who are, they are crocodile, right? So now again the pyramid is upright because at every trophic level the number of the organism is reducing. Please make these two.
Okay. Done.
Done. Okay. Now again we are only studying the number of oh, look, now come to here. We are studying over here this one. Next is this. Another example. We are just studying the number of pyramid only. Now, when we talk about the number of pyramid here in forest or tree ecosystem, now this will be D-shaped, irregular, right? Why? There is a single large banyan tree, but those which are eating fruit and leaves, they are deer, elephant, and birds. The number of the tree is just one, but its volume is very large. Here, but the dependent, those which are dependent on producer, their number are more birds, number of deer. So now this is bigger one, right? Then those which are dependent on deer, deer and birds like snake and wolf, their number will be less. Then those which can eat the wolf and all that, they're like lion, their number will be lesser one. So first the base is very narrow, then it become wide. So when you talk about the number of the pyramid in terms of forest or tree ecosystem, it will be irregular. Clear?
Okay. What will be irregular? The number of each?
The shape of pyramid is irregular.
Okay.
Right? This is irregular because it should be wide in one, right? The base should be wide, but it's not that. Why? Because the number of the tree is just very small.
Yeah, so on one tree there are lots of birds. Is that why it's
Right.
irregular?
For example, there is a single banyan tree, but the birds, there are 100 birds which are dependent on this tree. And the snake which visit to eat the bird, only two. Or wolf, but number of lion is just one. So this is D-shaped. Means irregular.
Okay.
Parasite ecosystem. There is a single tree. Who are the parasite on the tree? Because they are using the space, their body. Birds. So number of birds are more. So tree is just one, but number of birds. On the birds, there are parasite like ticks and mites. Their numbers are much more. Again,
Yeah.
on the parasite, there live some bacteria and fungi. Their number are much more. So when we talk about the parasite ecosystem, or pyramid of number for parasite ecosystem, it become inverted, right? Is not it?
Okay, so on top there's bacteria and what?
Bacteria and fungi.
Okay. And then there's parasite and what?
Parasite, ticks, mites.
Okay. Okay.
Right. Okay. So now in pyramid of number, if you want, you can write it down. In pyramid of number, we always count. Note it down. In pyramid of number, we always count the number of individual. We always count the number of individual present at every trophic level. Present at every trophic level.
In pyramid of number, we always count what?
Count number of the organism,
Okay.
present at every trophic level.
Okay.
Right? Fine. So pyramid of biomass. Right? Now this is pyramid of biomass. When we talk about the pyramid of biomass, here we take the gram dry weight per unit area. What is that? Means suppose that there is a 1 meter square area. In 1 meter square area, there is 100 kilogram grass, green grass. It doesn't mean that that is the biomass is 100 kilogram. What we have to do, we have to dehydrate the grass, because 90% is water over there. We have to dehydrate the water. Whatever the dry content will be, that we will consider only that mass. So when I say biomass, it means gram dry weight mass per unit area. Did you got that?
Yeah. So you take the dry mass of each.
Exactly. So please note it down.
Okay.
In pyramid of biomass, in pyramid of biomass, we always take dry weight. We always take dry weight per unit area. Dry weight per unit area present at every trophic level. So here we are taking the biomass. Look, first pyramid is grassland. Again, we are taking the grassland. Pyramid will be upright. Why? Because look, when we take grasses, the biomass of the grass will be more. The next, which are dependent on the grasses, they are the rabbit, their biomass is less. Then the snake, their biomass will be lesser. Then the eagle, their biomass will be lesser. Right? So the pyramid will be erect, or shape of the pyramid will be erect or upright. Clear? Is
Yes.
not enough?
Should I draw all of these?
Sorry?
Should I draw both of these?
Yeah. Please draw both of them.
Done.
Done? Fine. Okay. Now come to the pond ecosystem, which was erect in where? Pyramid of number. But in when we talk about the pyramid of this one, uh, pond, right? When we talk about the pyramid of biomass, it will become inverted. Why? The number of the zooplankton is much more, but their mass is very less. Those small fishes which are eating the zooplankton, their masses is much more. And large fishes, their mass is much more, right? So it will become inverted. Pond ecosystem, it will be erect in a pyramid of number, but it will become inverted in pyramid of biomass.
Okay. Done.
Done? Okay. Now, last but not the least, pyramid of energy. It will always be erect. Because why? Because 10% because of 10% law. At every trophic level, energy will reduce, right? Energy will never. So the perfect pyramid will be pyramid of energy. Clear? Please note it down.
Yes.
Pyramid of energy will always be erect. Pyramid of energy will always be erect at every ecosystem. At every ecosystem. Because at every next trophic level, because at every next trophic level of food chain, it will go down. So this was all about the pyramid of
Sir,
pyramids.
can you repeat it again?
Sorry.
Can you repeat it again?
So what do you written?
And till every ecosystem will always be erect
At every
at every.
ecosystem, the pyramid of energy will always be upright because at next trophic level of food chain, next trophic level of food chain, the energy will go down or reduces. Energy goes down or reduces. Clear?
Yes.
Now, what is next? Look, pyramid of biomass, we have studied. Pyramid of number, we have studied. Pyramid of energy, I told you. Now, what are the losses of food chain? Look, what is the losses at every trophic level? The energy is lost at during the respiration, like food burn. It goes at waste, means excretory system, right? So that is given over here, right? What is the primary production? What is the secondary production? Primary production, look,
Like
like the plant capture the solar energy and the 5 kilogram mass produced. So that is the primary production. I ate complete that 5 kilogram plant, but my mass increases only by 100 gram. So what is the secondary production? 100 gram. Got it?
Yeah.
That is the primary production and secondary production. Ask me if you have any doubt till this point, because in next class we'll start from here. Net primary productivity. What is the NPP? What is the CPP? Yes.
No,
Do you want to write it down? What is the primary production? What is the secondary production?
I can.
You can? Okay, fine. So that's it. So next class we'll start from here. Remember, or if you want to note it down in your notebook, we'll start from the net primary productivity. I will tell you what is NPP, what is ZPP, and all that. Right? Clear?